Hi, I'm Charles Wyckoff, and it's an absolute privilege to be here with a good friend and colleague, David Zacks, MD, Ph.D., professor of ophthalmology at the Kellogg Eye Center within the University of Michigan, and then co-founder and chief scientific officer at ONL Therapeutics. David, thanks for being on today.
Oh, it's an absolute pleasure. Thank you so much for having me.
But David, you've been a mentor of mine for years, probably in more ways than you've realized. You know, I've always looked up to you as both an MD and a PhD. There's a lot of us in retina, but you've really towed the line there from both the MD and PhD perspective, right? You're a practicing ophthalmologist, but also you have this passion that you've carried through for years in basic science. And maybe the question I'd start with is, what got you interested in the cell death pathway and retinal diseases originally?
Yeah, you know, that's really hitting right to the core of what we study and what we do. You know, as clinicians, we see a lot of patients, and I think one of the most heart-wrenching experiences, and I'm sure you know this, and a lot of the listeners can relate, is when you tell a patient that I'm sorry, but there's nothing more that I can do, and it really it's painful. And as we think about that, you know, and as we try to do something about that, and not have to say that as much, we realize that one of the core and key reasons people lose vision is because cells in the retina die. And my goal academically was to understand that process. And if we could understand that process at the molecular level, then we could potentially develop use rational approach to developing a therapeutic to try to prevent that cell death. By keeping cells alive, you know, presumably then improve visual function and outcome. So that really was the passion. And the reason for entering into that field.
That makes sense. And there's a lot of ways that cells die, right? I mean, there's sometimes non-specific death, and there's programmed cell death. So just at a high level, give us sort of how retina cells die. What are the different pathways?
Yeah, so you're absolutely right. Cells can die by pure necrosis. So, you know, we think of that in the setting of, you know, fulminant endophthalmitis, a big infection. You know, it's the atom bomb being dropped on the cells, and probably not much that can be done in that case. But in a lot of the diseases that we're talking about, AMD, glaucoma, which I consider a retinal disease, you know, retinal detachment, these cases, you know, you don't have that process going on of necrosis, but you have the activation of cell death pathways. And you're absolutely right. There are lots of different cell pathways, and there are probably more that are going to be discovered, you know, as we move forward. But the key to this and to the studying of this was to try to understand which pathways are activated in the context of this disease that we're studying. So I first started looking at this in a simple model of retinal detachment. And the reason I chose that was because it was an acute disease. It was otherwise normal, healthy tissue that was just suffering from an anatomic separation and an acute alteration of the cellular homeostasis. And then we just basically did biochemistry, molecular biology, probing and you know, interrogating the different death pathways, at least that were known at that time, and you know, seeing which ones were activated, and if so, to what extent. And from that, we kind of realized that, at least in the context of retinal detachment, the key regulator and the central regulator of photoreceptor cell death was the activation of the FAS receptor. And FAS receptor is a very upstream activator of cell death. And then subsequent to that, you know, and at that time, what we really knew about was apoptosis. Right. But since then, you know, we've, and the work of other people, a big shout out to Demetrius Vavas, who's worked a lot on this as well, you know, looking at necroptosis and other OSIS pathways that get activated. You know, it turns out that the FAS receptor is an upstream activator of all of these. And what really was interesting was that, and probably I didn't appreciate early on, was that the activation of this pathway was not just leading to cell death, but was also leading to the activation of the inflammatory cascade. The activation, the upregulation and release of chemokines and cytokines that activated then the microglial cells and the inflammatory milieu. You know, we don't typically think of retinal detachment as an inflammatory disease.
Right, right.
It's not, you know, you don't see cell, you don't see
No.
flare typically. It's
Yeah.
not a uveitis, but when you look at and you measure the cytokines in the vitreous, I mean, they are sky high. There's almost like a cytokine storm going on.
Hmm.
And when we block with FAS, we modulate that process. We modulate and we block the activation of cell death pathways, and we block and modulate the activation of the inflammatory cascade. And we think this inflammatory cascade serves as a feed-forward loop of inflammation leading to more cell death, and these inflammatory cells present more FAS ligand and get more cell death, et cetera.
So I'm going to ask you one deeper question related to fast signaling. So what's activating that fast signaling pathway? Is there some cytokine that starts the whole process off? Is it what's the initiating event that drives fast signaling?
That's still not 100% known, and that's something that we're looking at the academic level. You know,
right.
It's not unreasonable to think that perhaps hypoxia, you know, the photoreceptors are nourished. They get most of their nutrients, including oxygen, from the choroid through the RPE, physical separation. It could just be other stress active, you know, pathways, kinases. So we are actively looking at that at the academic level. But what we know, though, from an interventional point of view, is that you can block that pathway and have meaningful impact in terms of reducing cell death.
Okay, so you characterize fast signaling. It turns out it's actually important for many retinal diseases for cell death. And then you develop this molecule ONL 1204, right? So tell us about that development. Where did the idea come from to block this, and how did you come up with this molecule?
Right, so when we started first to look at the FAS receptor, we blocked it in lots of different ways. We used genetic models, we used siRNA against the FAS transcript, we used neutralizing antibodies, and we were just basically using kind of lots of standard different ways that you would biologically manipulate a system. And then a very interesting paper came out in Nature that showed in a completely different system that they could use a small 12 amino acid peptide to block hepatocyte cell death in an acute injury of hepatocyte toxicity. And it turns out that hepatocytes in their model, it died FAS-mediated cell death. And and this molecule was a fragment called MET12, which was derived from the MET oncogene. And you think, what does the liver and the MET oncogene have to do with the eye? So it turns out that the MET oncogene has a domain in it which sits in and blocks the FAS receptor. It blocks cell death, keeping cells alive and turning them into cancers. And this group in Pittsburgh, kind of found the core 12 amino acids in that alpha MET sequence and they called it MET12, and that was what was blocking cell death in the hepatocytes. The problem that they had was that they couldn't deliver it in a therapeutic manner because these 12 amino acid peptides get broken down very quickly in the bloodstream. Right. But we said, hey, let's just synthesize some of this. You know, it's 12 amino acids. They published the sequence. We could then stick it in the eye, and we did. And lo and behold, it works. It works as just like the other techniques that we were trying, neutralizing antibodies, et cetera. And we were, saving a lot of vision in rodents in models of retinal detachment.
First step.
And then I kind of had this aha moment, and there was a patient with a MAC-off RD who had their surgery delayed for a number of reasons, cardiac issues, et cetera. And while those were getting sorted out, I was working in the lab and injecting some rodents. We knew this was going to work. We had proven this over and over again. We were just getting the sample size up for publication and for validating it. And it just dawned on me, why am I injecting this just in an animal? I should be putting this in this patient who has surgery delayed, cells are dying.
Absolutely.
And that's when it hit me. And then,
yeah,
but there's a process, and they teach us a lot about medicine in medical school, but they never teach us where drugs come from.
Right.
And so, we were using this as a reagent grade product. And
when was that? What was the time? What year was that?
That was about, oh boy, I had a lot of hair then. I was looking, no, it was probably 2007, 8 in that range.
Wow.
And so it's been a little while, but then, working with the Office of Tech Transfer, you can't just take a product that's dissolved in DMSO. And then, we started, slowly working. We got some initial grants. So we formed a small company, and that was mostly just as a mechanism to do the development work. I never fully understood before that there's research and then there's development.
Right.
And, I thought they were all just one and the same, but it turns out, research is where you do the basic discovery. And that's really what the academic lab is about. And then development, that's what companies do for the most part. And so we had a small little company that we started, a couple of co-founders and I, and we got some funding, initially SBIR grants, and we were testing this out, showing feasibility. And then, but one problem that we had was, this MET12 was, we couldn't formulate it in a solution that would be compatible with the eye. So then we went on, a big drug discovery campaign, doing all sorts of analogs and manipulations. And from that, ONL 1204 was born.
Right.
And that's, we were actually screening for the ability to formulate it, but it turns out it was also a little bit more potent. And that became the lead product.
Impressive. So we'll make a very long story short. Now we'll fast forward now and say now we're in the clinic, because you are in the clinic.
Yeah.
So tell us about the ongoing first in human trial that you have going on. What data do you have so far, and what's next?
Yeah. So it's been really just amazing process to even get it into a form that you can inject into a human eye. And that really was the pivotal moment in the life of this project and of the company.
Mm-hmm.
You had a vial filled with approved drug, and you can approved for clinical trial use.
Yeah.
And very brave patients. So this is a first in human study. It's being conducted in Australia, and the first patient was injected at the Safe Site Institute in Sydney, Australia. And the PI there is Matt Simonovich. The PI at the Melbourne site is Penny Allen. And we've recently opened up a couple more sites. And the patients, because this is first in human, we want to take patients who had the worst prognosis. So these are patients with macula off detachment of greater than 7-day duration. So they typically do poorly. Or poorer. You can anatomically, put the retina back in position, but the visual outcomes on average are not as good as we would like. And the exciting thing we're seeing. So this, of course, is a safety study. It's not powered for efficacy, but we're very pleased that we're moving up the dose escalation, and so far safety looks really good. Tolerated very well. Doesn't seem to affect the surgical outcome at all. We're starting to get some hints as to, is this working in the eye? Without going into all the detail and boring everybody about this, but really we can probe that vitreous, because it's a surgical disease. We can take the vitreous out, and we can look at the activation of cytokines, their levels. We can measure those. And I would just say, it's looking positive, and it's helping us understand that yes, we are actually changing the milieu within the eye, and hopefully for the better. So that's really exciting. And now as we move the company forward with new funding that's wrapping up now, we'll be able to initiate new trials and expand the indications in which we're going to be testing this drug.
Phenomenal, David. That's super exciting. I mean, to have something in clinical trials that may actually stop or slow down cell death in retinal detachment would be powerful. And then thought that could be applicable to other retinal diseases. Really exciting, awesome work. Can I then probe sort of the sort of dynamics in your sort of professional life? So now you've got this role of chief scientific officer with a trial program that's very active with a lot of ongoing moving parts, I'm sure on a daily basis, and it's growing and expanding to other indications. How do you balance? How do you balance your time between seeing patients, your university, appointment, your colleagues, your residents and fellows that you teach, and then the CSO position? What's your day look like?
Yeah, so it's really about building teams. So first, for full disclosure, I'm a 100% employee of the University of Michigan. My relationship with ONL Therapeutics is managed by the Conflict of Interest Office, and I say that proudly. Everything is above board, and we want it to be that way.
Yeah.
And I have to really give a huge thanks to the University of Michigan, to the my former chair, Paul Lichter, and now my current chair, Paul Lee, to the Retina Service. It's really about building teams. I have been, just really blessed with fantastic colleagues at the Eye Center. We're busy, but it's a fantastic place to work clinically. The patients inspire me. My colleagues inspire me. My coworkers inspire me. And I think you and I are like-minded in this sense. We draw inspiration from working with fantastic people.
Yeah.
And then at ONL, I've just been really fortunate to have really good people, leadership there that has moved the company forward. I'm now a small cog in that machine. That's really taking on a life of its own. I work at ONL in a consultant capacity.
Right.
Again, through the University of Michigan, which allows that relationship, and I think it's ideal. And there are firewalls which are good and important to have, so that these conflicts of interest are managed appropriately.
Well, humble as always, David. And my last question for you then is: so we have a lot of people in retina that have made this transition, either full-time or part-time or on a consulting basis like you at this stage in your career, between sort of academics or private practice and industry. I mean, it's really a common theme across retina specialists, and it's really impressive for me and inspirational for me to see this across our space. What advice do you have? You've done this so successfully over the last 10 years. You've been able to really sort of bring value to the development space here, and yet still have a full-time position at an amazing academic institution. What advice do you have to people that are currently practicing but are interested in pursuing a relationship with industry or pursuing their own ideas from a development perspective?
I think I can really only answer as to what my path has been, and I think the core is to be true to yourself and ask interesting questions, and then you find the right people to help you answer those questions. I don't think there's, I think that, let me phrase it differently. I think that some of these walls we've placed in our minds are somewhat artificial. Look for the best people wherever they may be. They may be in an industry role. They may be in an academic center. They may be in a private practice. They just find the right people to talk to, and then you can, the conflicts. We all have conflicts in one way or another. It's about managing the conflicts, and that's why I'm always very careful to disclose fully and let people know what those conflicts are. They can be the judge of that. I'll be honest, I've only lost money doing research, so it's not like I'm getting rich off this thing.
Yeah,
It's but it's by asking interesting questions, and I think that it's important to do that so that you don't stagnate. Ophthalmology selects
yeah,
for some of the brightest people in medicine.
yeah,
It's just a privilege that we have, and we have active minds. We want to be engaged, and ask good questions, meaningful questions. Don't worry about whether it's industry, whether it's academics. For me, this is just the path that's ended up being
no,
the, a good one that's worked for me, for my interests, and but there is no right or wrong. I think the key is to just be true to yourself and ask good questions. I don't know if that's helpful to anybody, but that's what I found.
no, that's great. David, you're an inspiration to all of us in the field. You have an amazing story. I appreciate you sharing it with us, and we look forward to watching it continue to unfold. I appreciate you joining me.
Well, thank you, and thanks for all your wisdom and guidance too. You've played a big part in this.